Good morning. I'm sorry that we were a couple of minutes late getting started. We had some technical difficulties here. But we are pleased to have all of you online, and we are also pleased to have all of you involved in the community phase of this challenge. And so what we're going to do this morning is pretty simple. We're going to start with just a quick discussion of what it means to be in the community phase and what we're trying to accomplish here. And then we're going to go right into all of your presentations. So I'm very excited to see all of your names here, to see all of the people who are involved. I've been communicating with each of you. We've been watching your progress over the last couple of months, and so I'm really excited to begin to interact with you. And so the community phase of RA is a new thing that we're doing with Dream Challenges. It's an experiment in itself, and we're excited to see how it goes. So just to start, see if I can get my slides to advance. Oh, there we go. Okay, so as all of you are in the team phase, you already know what the outcomes were. There were two questions that we were asking with regard to anti-TNF therapy in rheumatoid arthritis treatment. The first was whether you could predict change in disease activity score on a continuous scale. And then the second was whether you could categorize individuals who would be non-responsive. And so what we found was that we were able to predict, you were able to predict better than random, but actually the outcomes that we saw, the predictions that we saw are, while they're robustly better than predictions that used simple clinical algorithms, they're not a lot better. And this was really the case both with continuous variable and with categorical variable. And so what we really want to do now is to take a step back and ask whether there is meaningful contribution of genetic signal to predictions here, and if so, can we really categorize what that looks like? So that's the goal of the community phase. So it's going to operate, very similarly to the team phase, with the exception that we're asking you all to start to work together. The idea here is we'd really like to bring together our thoughts and our approaches and our ideas and see if we can really meaningfully assess contribution of genetic signal. We want to use all of your great ideas in terms of predictive modeling methodologies, and we also want to incorporate what we already know both about this disease and the treatment and about genetic contribution through the forum of prior knowledge. And so not only are we going to provide you with the opportunity to work with each other, but we are also going to provide you with expertise from the field, people who understand the field very well, understand where there may be genetic signal, and may help in modification of feature selection and the incorporation of prior knowledge. So the theory is we gave you each a chance to do this on your own, and we were pleased to see the outcomes of that. And now we're all going to see if we can work together, both you and us. So how's it going to work? Well, each of your teams are going to organize into groups, and the groups are going to have 9 weeks to work together to improve predictions. We are not specifying how big a group needs to be, so if you all want to work together as one group, fine. If you'd rather self-aggregate into several smaller groups, also fine. But we think it's important that you self-aggregate rather than us assigning you groups so that you are actively working with each of your partners in the field. And as I said, we're going to incorporate experts in the field. So today we're going to discuss methodological approaches. And we're very pleased to have Trey Eidecker with us today. I'll talk a little bit more about him in a second. But we are going to continue to have webinars to provide information for you. And so I'd like to announce that the next one is going to be at the same time, 2 weeks from today, where we will have Eli Stahl and Robert Plenge discussing sources of prior knowledge for feature selection and the use of polygenic models in anti-TNF therapy analysis. So as you probably know, Eli is a statistical geneticist at Mount Sinai, and Robert Plenge runs the pharmacogenetics group at Merck. And they were the source of the discovery data set that was used in this challenge, and they have published previously on it, and so they're probably the best possible resource for understanding genetics and genetic priors specifically to ask this question. And so we think it makes a lot of sense to have their knowledge incorporated into your efforts. We do plan to have additional webinars moving forward, and we have a bunch of ideas of what we think may be useful for you, but we would like to get your feedback. So stay tuned for forum discussions about what other sources of information may be useful in your modeling efforts moving forward. So as I said, the goal is to identify whether any group can robustly improve predictions beyond those provided by either clinical predictions or by those in the team phase. And so we're not just asking you to beat the best possible model in the team phase, but we are also asking you to beat the aggregate model of all of the team phase predictions. So we don't want to just incrementally do better than we did before, but we really want to think thoughtfully about whether there's additional information or approaches that can be used that can best what has happened so far. So we will be building an aggregate model based on your individual predictions, and that is the benchmark by which we want to do better. And as with the team phase, within this phase, we are asking the top group to we're inviting them to join us as co-authors on a manuscript describing the community phase. And it's not yet clear whether there will be a single manuscript describing both the team phase and the community phase or separate manuscripts. It's really going to depend on what the outcome of this community phase is. So we will keep you updated with that as we move forward. So the goal for today is really to introduce the teams to each other so that you have a better sense of how you may want to self-aggregate into groups. And so we're asking each team to describe themselves and their methods on this webinar. And then we're asking you each to learn about each other and ask questions if you'd like. So you are all on mute right now. What I will do is I will unmute each presenter to give their presentation. And then if you raise your hand, then during each presentation, we can unmute you to ask questions of each other. So we are going to ask you to self-aggregate into teams. As I said, I'd like you to send me a list of teams you'd be interested in being introduced to by tomorrow. And by next Tuesday, we need to know what your new group is and what the membership is, and we can get started then. So we have a moderator today. The moderator is Dr. Trey Adeker, who's the Division Chief of Medical Genetics at the University of California, San Diego, and is a pioneer in the field of systems and network biology. And I have with me here Stephen Friend, who is the president and founder of Sage Bionetworks, who can give you a quick introduction to Trey.
Before I go to introducing Trey, I want to say one quick thing. Please know how many of us, including myself, are appreciative of the fact that you are the pioneers. Your willingness to see how things could work and work in different ways, I think we'll look back on in future years. And to all those who are on the call that have begun to form a colony that can connect and try to get to some sort of collective intelligence, it's just kudos, thrilled. And I think in keeping with that, Trey, who I think is going to talk next, is someone who loves experiments, loves looking at things in different ways, and has the appropriate role of listening, giving some insights, and I hope, Trey, getting a chance to sort of witness, be a part of this approach. And so, Trey, if you're there, I think it's your time, and I'm going to pass it back to Laura.
Yeah, I'm here, Stephen. So thanks for that intro, Stephen, and thanks to Gustavo, Regina, and Stephen for inviting me to be the moderator of the next 7 or 8 presentations. Yeah, I'm looking forward to it, and I think my group certainly has had an avid interest in these kinds of inference challenges for quite some time. We didn't participate this year, so this is a great opportunity to be a part of it.
Great, thanks. So I think without further ado, let's get started with the team presentations. So the first presentation is the Guan Lab, who were the winners of both sub-challenges, the co-winner of the second one and the outright winner of the first. And so let me pull up their slides, and I will unmute Yuanfeng so that the presentation can begin. So hold while I sort out these technical details. Okay, you are unmuted. Please take it away.
Okay. Can you hear me?
Yeah, we can hear you perfectly.
Okay. Thank you very much for inviting us. So we really enjoyed it, and we appreciate the opportunity to talk about our. So next slide.
We are having some echo. I wonder if everyone's not talking to go on mute.
Is the echo still there?
Yes.
Oh.
Laura, are you putting yourself on mute?
Well, not this second, but I was on mute.
Is it better?
It's not. So if you could turn the sound down on your computer, that may help. Let me try it.
Turn the sound. Is it better?
Still there. So let's see. Everybody's on mute. Myself, Trey, and you. So
Maybe Trey?
Trey, can you go on mute?
I was muted. I will remute.
Okay. And I'm muting. Okay, try again. Actually, Yuanfeng, I think you're on mute now. We can't hear you.
Really?
Oh, there you are. You're back.
And there's no echo.
And that's perfect. Great. Okay, so please proceed. Thank you.
Okay. So I just want to thank the organizers for organizing such a great challenge, and we really enjoy the team phase, and we are looking forward to the challenge phase. So right now we're on slide 2. So selection of features. So we use 2 ways to select our features. The first one is we retrieved some snips from literature. This was done by Google, and we went through approximately the first 2 to 3 pages of the results. And the other thing that we did was to find out the snips from this training data. This was done through a very straightforward F-test between the dosage, delta dash, final dash, and the different SNPs. So this was calculated for specific drugs. As you know, P-Link also implement the same function. So here we just kind of reinvented the way of P-Link. So, but if we implemented it correctly, the results should come up the same as if we use an outside software. So for clinical information, we use the baseline death, age, gender, and MTX. Cohort and batch information were not used since the final test set is in another cohort. Next slide. So our method is pretty straightforward. So a major thought in a major kind of motivation of our method is that we thought that it is unlikely that all the training samples are relevant. For each patient, it's more likely that only a subset of the training samples is relevant. This kind of model is in literature is actually considered pretty intensively, that you should find out the similar person instead of using a global regression or classification model. So to solve this problem, we use this Gaussian process regression model, which is the base learner to satisfy the criteria that I just said. So if you go through our code, there is some tuning of different ways between the clinical information and the SNPs. But this is really straightforward. So we can go to next slide. So we performed, I guess, pretty okay on the leaderboard data and also the final test set in terms of accuracy. And in terms of speed, right now it takes around 1 minute to finish the 600 to 700 predictions on the leaderboard data. This is not very satisfactory, but I think there's quite a big space to improve in speed. So next slide. So this is our team, Fanju, my postdoc, and myself. That's it.
Hey, Yuanfang, this is Trey here. So
yeah.
I've been really impressed by your method this year and also in the previous years. As you point out in your last slide, you guys have done consistently very well, not just in this competition, but in other competitions in previous years. What do you think is the secret sauce that makes, because the way you describe it, it's very clear, and you have a regression approach with input features. So what do you think distinguishes your approach from others? Because clearly there is a difference.
I think your question can be subdivided into two. So in general, I think I did pretty well in the, we did pretty well in previous competitions, not just these two dreams, but previous function prediction competitions, is that the model that we usually use are pretty simple. So it avoids some of the overfitting problems that otherwise would be seen. And another, I wouldn't say it's secret, but as you can see, usually for each challenge, it's usually when I was in graduate school, it's basically just me. So and now it's just the Fanju and me. So I guess there's a pretty low communication cost in generating those algorithms. So I think that actually works well for us. I think your question, if you, the second part of the question, I think if you particularly you refer to this model, I think again it's not secret anymore, but you have several cohorts in this data. So and the final test set is totally a different cohort. That means that almost directly means that a regular regression method that takes in account of all cohorts at the same time, instead of considering kind of local regression, is not going to work as well. So Does that answer your question?
Yeah, I'm not sure I fully understood the last part of your answer. I mean, I certainly understand the overfitting argument makes perfect sense, especially if you have multiple cohorts. But could you, so the very last thing you said, could you just repeat that? I'm just not, maybe it was just a connection. I just didn't quite get it.
Oh, so what I mean is, so for this task, we can try SVM, right? Or we can try some linear regression, but those kind of learning is over the whole spectrum. So if you have several cohorts, what you will regress out is the difference between cohorts.
Yeah, I see. So it's the Gaussian, it's the Gaussian kernel aspect that
yeah,
I see.
In terms of Gaussian, it's kind of local, so that would work better. It's just like
yeah. That makes sense.
Yeah, I was thinking the KN probably will just work as well as the Gaussian.
Okay, well, thanks. That answers my question.
Okay.
So Regina, how do we do this? Are others also allowed to ask questions, or am I the one? Regina?
Oh, I heard you, but I don't know what the organizers would say.
Lara, you are in mute.
I'm sorry, I apologize. I was on mute. Can you hear me now?
Yes.
Okay. So if anybody else has a question, please raise your hand and we'll answer it. Okay, so I see there's a question from Eric Kramer. Eric, I'm going to unmute you so you can ask your question.
And mute everyone else. Yeah, did you try other kernel methods or just the Gaussian process?
Oh, we tried many methods. So this one performed the best. And actually, in terms of leaderboard, we have a pretty big space to further improve the algorithm. But we kind of feel first, it's not necessary anymore, and second, it's going to make the model. Right now, it's pretty elegant since it's a pretty simple method. If I aggregate several, it will look less elegant for the final write-up. So we just decided to use this one.
Okay, great. Let's move on to the second presentation. Thank you so much, Feng. We really appreciated that. Let's see. The second presentation is Daniel Hidry, who's representing Team Outliers. So let me figure out how to make this bigger, full screen. And then let me unmute Daniel. Where are you? There you are. Okay, you are unmuted.
So you can hear me?
Yes.
So my name is Daniel, and I'm part of the Outliers team, which is a collaboration between people from the University of Toronto and SickKids. And I'll go through our approach to the RA challenge. So could we go to the next slide? Okay, so there's a couple of pre-processing steps that we performed. First, we restricted the SNPs that we used to ones that only had gene annotations, and this made it easier for us to, say, consider gene interactions. That's something that we did in a later step. Next, we removed SNPs which have info scores less than 0.9, and that was to avoid imputation biases that determined the cohort group just because of the imputation. And finally, we used genotype probability data instead of dosages because it allowed for more flexible models. Could we go to the next slide? Okay, for first, we built a linear clinical variable model to predict delta DAS. We use baseline DAS, batch and cohort, that's supposed to be drug, and gender. And some of the data was missing, so we imputed it with random forests and encoded categorical variables as indicator variables. So we built our linear model based on the clinical variables, and then what we wanted to do is find SNPs that could predict the residual information from the linear model so that they could provide us with more information than what was in the clinical variables. Next slide, please. Okay, so with these residuals, we had a third of the data, and we tried to, so we already had SNPs annotated to genes. So for each gene, we used all of the SNPs associated with it and tried to predict the residual delta DAS using a ridge regression model. And based on the correlation scores that we got, we ranked the genes, and then we selected a group of genes which have a relatively high ranking in our list and are known to interact, and this interaction was determined through an online gene ontology enrichment analysis tool. So we selected the top group of genes in our list that was selected from this tool. So next slide, please. Okay, so we already have a linear model relating the clinical variables to delta DAS, and now we can predict the residuals based on all of the SNPs that were associated with those selected genes. And so we performed both of these predictions for the Corona test set and added them together, and we used that for our final prediction. We also tried a lot of other things, and one thing that wasn't shown here is we had a method that was performing at second place during the competition in the Sub-Challenge 2. It involved combining both the clinical and genotype data through an unsupervised fusion method, and then finding variables that could account for most of the information represented in this fused representation and making predictions. But we made an error in submitting in our submission with the clinical ID or the patient ID names, so that's why we weren't able to see any scores. Okay, so I think that's it.
Yeah, so Trey here again. So can you talk about how you decided to, so you have these multiple rounds, right, I suppose, nestings of cross-validation. You have the sort of, you know, almost sort of 1-2 cross-validation implicitly that you're performing when, you know, at the very end when you perform on the unseen 2/3 of the data. And then within this approach, I think you said tenfold cross-validation on, which I guess is on the third of the data. Is that right?
Yes.
I was just a little unclear on how this, what the logic is behind the cross-validation.
Well, we wanted to evaluate which genes most predictive. So we had to do validation because you're doing a predictive model. And then 2/3 of we wanted to also. Oh, I see what you're. Okay, sorry.
Well, so basically, you have like a 1-2 cross-validation framework for the final model, right? So when you hide 2/3 of the data, right?
Yeah.
And then if you go back a slide or two, I'm not sure if we can do that. I think during the variable selection phase, you had another cross-validation step, I believe, right?
So during variable selection, we were just ranking the gene. So to make that rank, we needed to evaluate the performance. Is that clear?
Yeah, yeah, sorry, the connection's pretty bad. I'm getting like every other word.
Yeah,
I don't know how my voice is.
Trey, your voice is great, but when you are off, everybody else echoes. So don't.
I mean, I'm kind of getting it though. So so if we advance the slide again, maybe I just missed something. So it's basically a 1-2 cross validate. Oh, there it is. There it is. So then step 6. Could you just explain a bit better what happened in step 6?
So we have one data. We split it into 10 parts. Then we, with 9 of those parts, we build a model for each gene and evaluate how well it does on the 10th part. So we do that for each of the folds or parts, and we get an average correlation score. So now we have a ranking for the genes. Then if we go to step 8, we're going to select a group of genes that interact and have a relatively high ranking. And then with the remaining 2/3 of the data, we build a model to predict the residuals.
Yeah, so another interesting aspect was step 8. Here you've essentially looked for genes that are in a similar pathway. That's what step 8 is doing, right? Of all the features you could select, you bias it to select features in the same pathway. Philosophically, one might have chosen the exact opposite, which would be to get a representative gene from each of, say, some number of top pathways. How did you think about choosing essentially genes from the same pathway versus representatives from multiple pathways? Did you try the other?
So we did not try the other. The reasoning for choosing genes in the same pathway, since all of these genes had a relatively high ranking, then we would think that this pathway is important, and changes in the genes based on those SNPs and any of those genes in that pathway might have an effect on the response.
Yeah, and it seems to be working. Yeah, I mean, maybe what's happening is any one gene is a noisy predictor of that pathway activity, but somehow the aggregate is a very robust predictor of that pathway activity or something like that.
Maybe.
Yeah. All right, thank you.
All right. Yeah, thank you very much, Daniel. Let's move on to the third presentation, which is Team MI, which is represented by Liu Cheng. So I will now pull this up and unmute Liu. Okay, you are unmuted.
Okay, can you hear me?
Yes.
Okay, this is Team MI from Finland. So our team members are from Helsinki Institute of Information Technology, called HEAT, and also another part of the team is coming from Institute for Molecular Medicine Finland, which is called FIM. So team members from HEAT are basically doing research in machine learning, and then team members from FIM are doing research in genetics and also biology. So could you move to the next slide? So let me first tell our findings and SNP sets. So we found that the clinical variables or covariates are very predictive. So this means that the phenotype is strongly associated with a covariate. This will mean that a predictive SNP in the GWAS sense that will inevitably be correlated with a covariate. So now the feature selection is a key part in this challenge. So we propose the following criterion for selecting the features. So features should provide independent prediction power on top of the covariate. In other words, the combined prediction of the genetics and covariate should be better than the covariate only prediction. So, based on this criterion, 7 SNP sets are selected. So there are 463 SNPs in total, and these SNPs are selected from the literature and give us a different differential gene expression. We also did some other experiment like pathway selection, but they didn't work very well. And these SNP sets provided additional prediction power on top of the covariate, both in sub-challenge 1 and 2. So it has been validated on 3 independent data. So in the cross-validation phase of the training data and then the leaderboard data and the corona data. Also, we observed the same phenomenon on both of using both of our methods. So we are going to we expect that these SNPs will also work for other methods. Could you go to the next slide? So we used two models. We have tried several methods, and then we introduced 2 most successful ones. So 1 linear and 1 nonlinear one. So the first method is Gamma, which is an implementation of a linear mixture model. So directly using the Gamma use of the SNPs doesn't really provide a good prediction result because we observe that the covariate dependent component of a predictive SNP is really very large. So in overall, this will overweight the covariate in the final prediction. So what we did is like we extracted the covariate independent component for the prediction, and the result is 3 and 8 in subchannel 1 and 2 respectively in the final corona data. Of course, this model is not perfect. It's a really simple model, and it's based on simple assumption. So the disadvantage is like all samples are assigned the same weight, so all patients are treated equally in the prediction. And the underlying model may be nonlinear according to the current results. We feel like it's highly likely it's nonlinear instead of linear. So could you go to the next slide? The next method is we call it a BMKL. So it's a Bayesian efficient multiple kernel learning method. So it's a top performing model in DREAMM-7. As we can see from the formulas in the lower part of the slide, so it is a very flexible method. This K is like a kernel, which is a similarity matrix, and it allows it integrates different kernels using different weights, and also different patient can be assigned different weights, which we call the patient weight here. So this is a powerful nonlinear method. So in essence, this method is like very similar to Gaussian process regression. So the kernel here is actually the covariance matrix in the Gaussian process. However, the result is not as good as a linear model. So the rank is 12 and 9 in subchallenge 1 and subchallenge 1 and 2 respectively. After seeing the leaderboard results of the corona data, we feel that we may overfit the model, and we believe it will perform better once we reparameterize the model. So could you go to the next slide? So we are not really sure about how the collaboration works, but we welcome any kind of collaboration. So if you feel our SNPs or our method are useful, please contact us. Although the organizer is going to build an ensemble predictor, we are also highly interested in building a separate ensemble predictor that combines existing models and even coordinating the process. Ensemble predictor is in general better than any individual predictor. We have experts who have experience with this. To advertise ourselves, so we have a complementary expertise to the winning teams. So we have informative and reliable SNPs, and also we have a powerful kernel method to improve. Also, finally, we want to mention that we have new expertise to select the SNPs, and using the data currently we are running new experiments. Thank you.
So this is Trey here. So can we talk a bit more about how you selected the informative and reliable SNPs? I think that was maybe the first or second slide.
Yes. So basically, we combine for any we combine the SNPs and also the covariate for the prediction. And we already know the prediction for the we already know the prediction using only the covariate. So if the new prediction, if the combined prediction is better than the covariate, then we think that this is a good SNP set.
By better, does that mean you're training to predict the residuals after regressing against the clinical covariates?
Pardon?
So just technically, what did you do? Did you first train a model to predict the outcome based on the clinical covariates, and then, like previous groups described, you then regress against the residuals?
Yeah, that's correct.
And then what I thought was an interesting feature, so the 463 SNPs, those you selected using prior knowledge before you looked at any data at all. Is that right?
Yes, that's.
Yeah, so that's really interesting that you basically the entire procedure only revolved around 463 pre-selected SNPs.
Yeah.
Huh. Any other questions?
Oh, we do. We have a question from Daniel. Daniel, I'll unmute you now. There you
go. You did SNP selection with GWAS. Was the GWAS selection using the data that we had?
Yes, the GWAS is using the data we have.
Okay, thank you.
Did you do the GWAS yourself, or did you use the results reported in the literature from this data?
We did it by ourselves.
No. Okay, great. Well, thank you very much. I appreciate your presentation. Let's move on to the next presentation. Let's see. The next presentation is Eric Kramer representing Team STSI. So let's see, we'll get that going, and I will unmute Eric. Well, actually, you are already unmuted. So if you unmute yourself, actually, it looks like Eric's having some audio trouble. I think he's trying to call in by phone, actually. So I'll tell you what, let's move on to the next one, and then we'll come back to Eric as soon as he has sorted out his audio trouble. Okay, so the next is Team Tracy, which is going to be, oh no, wait, Eric's back. So hold on, Eric, I've unmuted you, are you there?
Yeah, sorry, I got disconnected right when you unmuted me.
Perfect. Okay, there you go.
Cool. So we're from the Scripps Research Institute in San Diego. Me and Bhuvan are two PhD students, and then Nate and Ali are faculty here at Scripps. Yeah, so the first thing we did a heritability estimate, and we found that it was pretty low. So we submitted a clinical-only model. So we just took these 5 clinical variables: baseline, disease score, age, gender, drug, methotrexate. We took that, if you can click forward. Yeah, we put that into a QBIS regression model. So this is a model made by Max Kuhn at Pfizer. It used to be proprietary, but it was open source a couple of years ago. So there's C code and R code to use it. Yeah, and then we just made predictions with just the clinical covariates, and we got a pretty good result. So that was our clinical only model that we went ahead and submitted. In addition to that, we submitted a genetics model. Yeah, so keep going. Yeah, so the first thing we did a multidimensional scaling followed by a clustering. So just kind of looking for population stratification. And what we see here, the green over there is actually one of the cohorts, the Bragg's cohort from the United Kingdom. And the red there is a bunch of Scandinavians. And so we were doing cross-validation on our model, and we actually never performed as good on the Bragg's cohort in green as we did on the red people, which seems to suggest that we're not accounting for this population stratification somehow. So yeah, if anybody has advice on that, that'd be great. Yeah, so that's the Bragg's cohort. Click again. Yeah, and then those are a bunch of Scandinavians. And keep going. Click, yeah. Anyways, so then our other input. So we took known rheumatoid arthritis and rheumatoid arthritis SNPs from the literature. We computed HLA types for all of the patients. So we used the HiBag package. It's basically a random forest that then uses that to predict HLA types. We computed likely deleterious mutations, so we used Polyphen, SIFT, and COMDEL, and then we aggregated those mutations to genes. And we actually did gene ontology and drug bank and all of those. So then we had cumulative scores for how many deleterious mutations each patient had in various genes and various gene sets. We also took the Wellcome Trust data for rheumatoid arthritis. We built a predictive model just for the presence and absence of rheumatoid arthritis, and then predicted on the people in this challenge. And so basically we found that their overall rheumatoid arthritis risk was correlated with their disease severity. So that was kind of interesting. And then we recalculated a GWAS meta-analysis on all the SNPs. But we couldn't find a good way to include these SNPs without overfitting our model. So in the end, we didn't include those. Yeah, and then here's kind of the output from, this is our importance. So it's kind of, sorry, the y-axis is importance like in a random forest. And you can see what's most important for our model. So of course, the most important is baseline disease severity, followed by the drug they were given, their age, and then 2 known SNPs from the literature. Gender, some more known SNPs, methotrexate, and then somewhere in here you can actually see some of the HLA types are coming through too. So the HLA types were used in the model. Yeah, that's pretty much all we did.
Hey, so interesting presentation. A question I have is then you getting back to this population substructure.
Yeah.
I'm getting an echo of myself. Are you guys getting that echo? I'll take that as a no. So yeah, so your good performance then is not on the green. You think your good performance is all coming from the red and maybe the blue? Is that the idea?
Yeah, and so yeah, that's what we're seeing. So basically we did cross-validation, and we found that we were doing worse in the green patients and much better on the red. We tried stratifying the models, so we actually built separate models for the Bragg's cohort and then everyone else, but for some reason that didn't seem to improve the model. So the final model we submitted wasn't stratified based on this clustering, but we thought it was pretty interesting and worth sharing. The corona patients, interestingly, kind of end up both with the green patients and with the blue patients. So the corona patients are kind of all over.
Unmuted. Okay, thanks. Other questions?
Well, I think that's an interesting point about the corona. About the corona people being showing up across both of those populations. Corona ascertainment, I believe, was entirely within the United States. So it's interesting it's showing up with the Scandinavians.
Yeah, I mean, I'm not that surprised just because of mixed ancestries in the United States as opposed to Europe, but yeah.
Yeah, fair enough. Okay, so it looks like we do have a question for you from Yang. So Yang, I'm going to unmute you so you can ask your question.
Okay, yeah, thank you very much for the very interesting presentation. may I know how do you use the treatment information in your model, the clinical-only model? Do you use the specific drug or use the group of drug? Because the training and the testing use the different drug combinations.
Yeah, so we just transformed the drug, not by a class of drug, by each individual drug. So we created dummy variables, just a 1 if they got a specific drug, 0 if they didn't. And so that was input into the model. And then we did a random forest imputation for all of the missing data. I saw that another group did that. So yeah, so any missing data we imputed with a random forest.
But I'm sorry. In the testing set, there's 2 new drugs. How do you predict the, how do you utilize those information?
Yeah, and so we looked through if there were new features like that, we deleted them and then re-imputed them using the random forest.
Okay, thank you.
Okay, thank you very much for the presentation. So we'll move on to the next presentation, which is Team Tracy, which is represented by Tao Fang. So let me unmute. Where are you? There you are. Okay, you are unmuted.
Hello, can you hear me?
Yes.
Oh, okay. So we are Team Tracy from Quantitative Biomedical Research Center in UT Southwestern. We have 4 members in the team. I'm Tao, the graduate student here, and the other 3 team members are now faculty members. Okay, please go to the next slide. Okay, so this is our rationale behind the prediction algorithm that we used. So we have 5 kinds of clinical covariates. We have a lot of genetic information. So those input data represent a heterogeneous set of input data that we could use for prediction. And also we have a lot of different prediction algorithms, common ones like support vector machines, random forest, AdaBoost. So those algorithms have their own advantages and disadvantages. So we think that maybe we should use one prediction algorithm to look at a subset of the input data to best utilize its advantage in considering those types of that subset of the input data, and then we make predictions using different prediction algorithms, and finally aggregate the prediction to give a final result. So this is our rationale behind, and please go to the next slide. So another thing that's worth mentioning is how we selected the genetic information. So we have like 4 subset of, or 4 layers of SNPs that were selected. We selected 20,000 immunology-related SNPs. We this is how we did it. We pulled out the CAG pathways and took out the genes that are members of immunology pathway. That gave us a few hundred genes, and then we look at the SNPs whether it is close to or within one of such genes, and that's how we selected the 20,000 immunology-related SNPs. And also we digged in, looked into the published literatures and find about 300 SNPs that are related to rheumatoid arthritis, or related to MTX response, or related to anti-TNF-alpha response. So we have these 4 layers of SNPs that were selected. And then, because we still have too many SNPs, we need to reduce the dimension. For all the SNPs and for the 20,000 immunology-related SNPs, we use PALINK to the MDS method within PALINK to reduce the dimension. And for the other 3 types of SNPs are SNPs we use just a principal component to reduce the dimension. And please go to the next slide. Okay, so we employed a very similar framework for sub-challenge 1 and sub-challenge 2. So in sub-challenge 1, we have 5 submodels. For the clinical variables, we used baseline DAS, age, gender, MTX as covariates. And for the drug, it's we use kind of a special method. We have 5 submodels. In submodel 1, 3, 5, 1, 3, 4, we use the drug information as a covariate. And in submodel 2 and submodel 5, we use the drug information to stratify the patients and make predictions in each of the 3 groups. And as for the genetic information, we use the SNPs or the principal components generated by MDS or principal components as covariates. And in each of the submodels, we used random forest, lasso, support vector machine, AdaBoost, and partial least squares. And we used cross-validation to assign different weights to the prediction results from the 5 submodels. And finally, you just use a linear combination to aggregate the prediction of results. And in subchallenge 1, another special thing we did is that we treat sertolizumab as adalimumab, and golimumab is treated as infliximab. We did this because we find a paper that said that is some meta-analysis, and they suggest that the 2 additional drugs that appeared in the test set is very similar to the 2 drugs adalimumab and infliximab in the training dataset that we see. And please go to the next slide. So in subchallenge 2, this is kind of our prediction algorithms is very similar. It's just we use cross-validation to adjust the weight or some parameters of the prediction algorithms, so to make it better to predict the binary response, and that's our method.
Okay, thank you. So on this last slide, you're listing 5 different approaches. Is that right?
Yeah.
Different algorithms. And which were the 2 that you submitted?
Pardon?
For the final test set, right? You were, for the corona data, you were able to submit 2 predictions. Which of the 2 are represented here? Oh, you did it's an aggregate of these. Is that right?
Yeah, aggregated of the 5 submodels.
Right, gotcha. Okay. We're running a little bit short on time, so I guess, Trey, if you have a burning question, please ask it. Oh, I have you on mute. Now I see why you're not. But otherwise, we should probably move on because I want to make sure all of the teams get a chance to fairly represent themselves here.
If it's okay, can I ask a question myself?
Yeah, that's fine. I was muted, but I can stay muted.
Hello?
Yes, please ask your question.
Oh yeah. So as you mentioned before the in the previous seminar, you said that you only used a subset of the final corona test set to score all the predictions. So may I know how many patients were actually used in the scoring phase?
Absolutely. I believe it was on the order of 600 or 700. So there are some additional samples that were genotyped, but for whom clinical data is not yet available because they're still being followed up. So I hope that the test set will actually increase in size for this community phase, although I don't think we should be expecting more than a 10% increase in size as they continue to collect clinical data throughout the summer.
Okay.
Okay, thank you very much. So let's move on. to the next presentation, which is the SBI Lab, and will be represented by Javier. So, okay, let me unmute Javier. There he is. Okay, you are unmuted.
Okay, hello. I'm Javier Garcia from the SBI Lab in Universitat Pompeu Fabra in Barcelona. Can you go to the next slide, please? So our team is from a lab of structural biology led by Professor Baldomero Oliva. And the team is composed by biologists and computer scientists like myself and Jaume Bonet, 4 biologists, computer scientist, a mathematician, and a statistician. Most of the participants are PhD students, except Daniel Aguilar and Joan Planas Iglesias that are PhDs. Yeah, can you go to the next slide, please? So, first steps we did in the project was we wanted to integrate as many data as possible, just to select the candidate SNPs. So we mapped SNPs to proteins using Biomart, and also the consequence of these SNPs. So if it's a synonymous mutation, if it's in an intron, in an exon, in frame deletion, etc. We also predicted the consequence of the SNPs, if it's tolerated or deleterious. We mapped all diseases known to be associated with each marker, also from databases, and also drugs known to target the proteins mapped to the SNPs using drug bank database. The idea was use this data to select SNPs, combined with in-house algorithms like gen prioritization algorithms using protein-protein interaction networks from a platform we developed, Diana, and using a gene prioritization algorithm called Gildify. Here is the URL address. And at the beginning, we use arthritis just as the keyword to generate the seed proteins, to generate the protein-protein interaction network. Then we tried also to use a variant using microarray expression data to modify the scores of the proteins in the network using based on the relevance in the microarray. And we also calculated association and correlation measures between marker dosages and arthritis response, both delta dash and response, non-response. So we combined all this information into a MySQL database just to have all the data. Can you go, please, to the next slide? Okay. So we divided, the project is divided in 2 sections. One is candidate marker selection, and then the prediction itself. At the end, the candidate marker selection, as I will explain in the next slide, was mainly based on association and correlation measures that were basically Fisher's exact test and Pearson correlation. We calculated this specifically for each drug because we observed that the markers for each drug we obtained were completely different. And as close markers tend to be linked, we just selected one marker for each protein, the one having the best p-value in the Fisher's exact test or the best Pearson correlation. So once we selected some markers for each drug, we tested different prediction methods. We used the software Weka, and we did the analysis using tenfold cross-validation in the training datasets. With the selected markers, we tested different classification algorithms like linear regression, supervector machines, decision trees, neural networks, Gaussian processes for regression, principal component analysis, etc. We also explored to predict delta dash or relative delta dash, to predict binary response, non-response. The main problem we had, as we will see in the next slide, was overfitting. At the end, the best combination that provided the better and more stable results between training and testing datasets and the leaderboard dataset was the usage of Gaussian processes for regression, the same as the used from the Team One lab. So we finished exactly with the same prediction algorithm and to predict delta dash. And we used exactly the same procedure for sub-challenge 1 and sub-challenge 2. The only difference is that for sub-challenge 2, that we had to predict response, non-response. So those with a delta dash close to 0 were predicted as non-responders. Yeah. Can you go to the next slide, please? So as I told before, the main problem was overfitting. Initially, candidate marker selection was done using multiple sources of information, as I said, gen prioritization algorithms, protein, drug data, etc. But we had very good results in the training data set and in the tenfold, but at the beginning we did several submissions to the leaderboard and it was very difficult to replicate these results. We were not able to replicate them. So we saw that there was a problem of overfitting. Then, just to simplify, we prioritized the candidate selection based on association correlation measures, but again, it was difficult to replicate. So at the end, we said, okay, let's see why we are not being able to replicate the result. So we just used the test data set to test which of our candidate markers were having some response in the leaderboard dataset. So we tested for about approximately 70 markers, and we were able to confirm that only about 20% of them were having some response in the leaderboard. So it indicated that our candidate marker selection has to be more stringent because there is a lot of noise. And yeah, can you go to next slide, please? So just what we are planning in the collaborative phase is that, okay, we have to improve our marker selection in both ways, to increase the list of candidate markers, because for each drug we have from 20 to 30 markers approximately. And so improve it just by increasing it or reducing it. We have not used GWAS analysis tools like Plink, so it would be interesting to collaborate with groups that know how to use these tools, or to use other marker selection criteria proposed by other groups, and also to improve the classifier performance. So that's all. Yeah, it's all.
Oh, hi. So Trey here. So I think my main question was to clarify, the very beginning of your talk, you presented available data where you had a lot of information about each SNPs, or each SNP in the study. But then it seems like, did you end up using that in the regression? The answer is no, right? You basically selected your features essentially strictly based on association score.
Yeah, the main idea was to use all this information, but after several submissions, we saw that there was overfitting, that we were not able to replicate these results. So just to simplify, we just used association measures just to select. It was a pity because it was in the training dataset there were promising results, but not replicable in the test dataset. So, and just to simplify, we selected just based on association measures.
Okay, thanks.
Thank you very much, Javier. All right, let's move on to the final presentation, which is going to be given by Matthias Samwald, who is representing Links to Open Data. Matthias, I am unmuting you now. There you go.
Okay, thanks a lot. Can you hear me well?
Yes, very well.
Great. Hello from Vienna, Austria. Probably here because Stephen Friend came to Vienna and gave a very inspiring talk. And yeah, just a few months have passed and here I am. So we are probably the most unusual participant in this team phase, and maybe a bit of a background. This team is actually international and was formed around the World Wide Web Consortium Semantic Web in Healthcare and Life Science Interest Group. So I'm from the Medical University of Vienna, and some other team members were from the University of Leipzig as well as Stanford University. Next slide, please. Next slide, please. Yeah. So in this World Wide Web Consortium group, we have for several years now worked on something called linked open data. which basically tries to use the philosophy that made the World Wide Web so successful to the data domain. Basically, and I don't want to go into too much detail, the idea here is to make as much data, and of course this also includes biomedical data, available in an interlinked and open format, in the hope that this also enables new insight and new collaborations to form. And in this World Wide Web Consortium group, we build up some expertise in how to use various linked open data or linked data standards, such as the resource description framework or the web ontology language, to enable collaboration and research in the biomedical sciences. Next slide, please. So what we did for this challenge, and we basically focused on sub-challenge 2, was to use data that is currently available in this cloud of linked open datasets on the web to create models for predicting drug response. So we used ontologies such as the gene ontology, of course, or drug bank, basically stuff that also some other teams used. But instead of going directly to the raw data, we made use of this interlinked representation that has been made available as part of this linked open data effort. So part of our work was not only to solve this challenge, but also to test how well these interlinked datasets and these open standards can be used for solving such a problem. And as you can see, we were able to write quite sophisticated queries that spanned across different datasets and that helped us to enrich the models we used for training our machine learning models. So basically, we wrote a few scripts that converted the challenge data, the patient data, both the clinical and the genetic data, to these interlinked formats, and then also pulled in interlinked data from other relevant biomedical data sets, and then ran a bunch of queries over the aggregate data set to see what kinds of insights we can get from that. Next slide, please. So basically, we tried two different machine learning approaches. One of them very unusual, I would think, and one of them quite simple and standard. So the first thing we tried was a software package that was developed by people on our team called DL Learner, which is a very flexible software package for inductive logic programming that basically can digest these linked data graphs and the ontologies that we were assembling for this challenge. So the basic idea was that if we can get a lot of data in one coherent model, and this is quite easy to do with the standards we used, this DL Learner software would be able to identify some properties of the patients that differentiated responders from non-responders. And these logic programming algorithms are quite flexible, so they are not just dealing with tabular data, but they can actually use richly structured hierarchies and relationships. And the classifiers you get from the software are basically based around logical axioms that tell you how the properties of, for example, the responders would differ from the properties of the non-responders. So in this specific example, I just copy-pasted such a resulting axiom from another test data set. So this has nothing to do with the current challenge, but it is a nice illustration. So here you can see the result that was generated by this DL Learner software for differentiating compounds in a test data set by their carcinogenic properties. So you can see that it's a richly structured logical axiom with existential qualifiers, etc., etc. So we tried that, and we got rather well, not so robust results, unfortunately. So we found that we didn't restrict the parameters enough, and yeah, the algorithm got a little bit lost in the large amount of data that we assembled. So there's still a lot to be optimized here. But luckily, we also tried a more simple and standard approach on the data we gathered. And for this, we simply used scikit-learn, a Python machine learning library that allows you to relatively easily run different machine learning algorithms over a data set. And yeah, we tried to use this also in our data. And after coming up with very different and complex solutions, we ended up with a strikingly simple solution, only involving 6 SNPs in total. One of them associated with TNF-alpha inhibitor response, and the 5 others associated with methotrexate response. I also used, looking at how the mechanisms are for these different SNPs, I also calculated based on 4 of these SNPs a so-called methotrexate score that yeah, improved the model a little bit. And we used a gradient boosting classifier in the end, simply because it performed best among all the classifiers we tried. Next slide, please. So what we hope to bring into this collaborative phase in terms of expertise is to represent the biomedical and pharmacogenomic background knowledge in a richly structured and interlinked way, and basically representing and querying this complex background knowledge as graphs and ontologies to try to find hidden patterns that were not really explored so far by other teams in the last phase of the challenge. Besides that, with the expertise on these inductive logic programming algorithms, we might also be able to come up with novel ways of using ontology-based classification to, for example, select those SNPs that might be relevant, or to find unusual combinations of SNPs and other patterns that might have been overlooked with more classical machine learning algorithms. Okay, that's it from my side.
So Matthias, this is Trey. Fascinating presentation, definitely a different approach. I was unclear on the last slide, what was the interaction between DL Learner and the final classification with the scikit library? So you say only 6 SNPs were used. Did any of those come out of, say, an ontology-based approach, or not? I just got confused as to how the top part of the slide relates to the bottom part of the slide.
Yeah, so it hardly relates to each other at all. So we, when trying to structure the data for the DL Learner, we also ran some queries and got some data out of this. But we had to conclude that with the approach we used in the team phase, provide any helpful results. So basically, the lower half of the of the slide is quite independent of from the upper half. So yeah, the scikit-learn models only are related because we had some made some experiences, but they are not related in any other way.
Okay, great, thanks.
Okay, well, I'd like to thank everybody for the time they put into this and for their presentations. And as we wrap up, Trey, I wonder if you would be willing to just provide some thoughts maybe in terms of what you've seen in terms of commonalities or differences or overlying themes across the various
Yes, absolutely, Laura. I was hoping you would ask. So I've been taking notes, and I'm sure some of us also, I'm not the only one, but the themes that emerge, I think, were at least threefold, and each of the 17 that presented touched on at least one of these themes, and sometimes multiple of these themes. The first is this trade-off between local and global regression. So we saw certainly the Guan Lab team made a big point about using the Gaussian regression, the local regression. I think we also heard the same point made by Team MI, by Lu Cheng, again, on the importance of local regression. And then I think Eric Kramer also talked about essentially how this data set had at least 3 very different cohorts. Again, that has to be dealt with. So the first theme, I think, is this theme of heterogeneity in the data in the form of at least 3 cohorts in this RA data set, but in general, we have to worry about it. So local regression would be theme 1. Theme 2 is pre-selecting your SNPs from prior knowledge. Some teams did not do this. Some teams relied heavily on prior knowledge before looking at the RA-related data at all. I think the strongest example of that was Tao Wang, who talked about essentially a large database of information they used to select SNPs before they even see any green data. And that was a fairly interesting idea. I think there's softer forms of this that a lot of groups use, which basically try to simplify the feature set as much as possible before ever exposing that feature set to the learning problem at hand. So that's theme 2. And in theme 3, that I think that was just underscored in this last talk, but also in some others, is this idea of applying prior knowledge not before the data, but applying prior knowledge during the learning procedure itself, either in the form of Go, gene ontologies, or pathway knowledge. Or I think the most interesting case of this was Eric Kramer's HLA type. So the idea is that that's a little bit like a pathway function, if you think about it. I was trying to work in HLA type into a sort of more general philosophy for how one goes about this, but I think it's maybe most similar to grouping SNPs in a pathway, or like this last talk by Matthias, applying some logic function to a group of features. So those are the 3 themes I saw, and I think those 3 themes do incorporate all of the 7 presentations we saw in at least one of those themes. So again, local versus global regression / dealing with different cohorts. 2, pre-selecting SNPs, or this decision of pre-selecting SNPs or not pre-selecting SNPs before the data are even seen. And then 3, using pathway knowledge or logic functions or other types of knowledge to group SNPs and apply constraints on SNPs during the learning process.
Yeah, I think those are all really important points. Things that need to be dealt with in these data set are obviously subpopulation differences and feature selection, and I think everybody has something to bring to that those questions, and there's some obvious synergies across the way that that people are approaching these. Trey, I just put you on mute for the purposes of feedback because I'd like to unmute Gustavo, who wants to make a couple of comments as well. So I'm going to do that. I'm sorry I can't have you both unmuted at the same time. The feedback, for some reason, makes that not tenable at the moment. We'll give it a try after Gustavo said a couple words. Gustavo, please.
Thank you. Thanks everybody for sharing your things that you learned during this challenge. And it's clear that there are many complementary and synergistic ways of thinking of this data. I want to add one thing to what Trey said, which is it seemed to me that one common theme is that simpler seems to be better, that the methods that were too ambitious in a way seem to have done a little bit less accurate predictions than the ones that were a little bit simpler and less ambitious. But I don't know whether this is a correct statement or whether it is that we haven't found the right way to think of it. But the other thing I wanted to say is that I was wondering how you guys, and probably this is a question for you, Lara, to move forward, is how can we then help people find each other so that they form teams, or how do you envision, Lara, this next move, which is that people start to work together in order to find the synergies? And thank you very much, everybody, and I will mute.
So what I'm asking is for each team to send me a list of the other teams they'd be interested in being introduced to, and then I'll ask each of you to start interacting with each other and come up with your groups on your own over the next week. We can certainly provide you with formats to interact with each other if you'd like to set up webinars or anything like that, although I recommend Google Hangout as a great way to do this on your own, which would make you a little bit more independent than needing to go through us. However, if we can help in any way to facilitate that, please let us know. More details will be forthcoming on exactly how we think that this will go. So the task right now is to form your teams or your groups. And then we're going to have another webinar 2 weeks from today in which we're going to start to talk about feature selection and polygenic modeling, as I said, with Eli Stahl and Robert Plenge, who are intimately knowledgeable about both this data set and the field in general, to see if we can help with the SNP selection and pathway approaches using their prior knowledge. So we'll see how it goes from there. So with that, I think I'd like to thank Trey for taking the time to join us today and his insight. We really appreciated having you today.
No, my pleasure, Laura. I hope it was helpful.
Absolutely. And I'd like to thank all of the panelists for taking the time to join us, to listen to each other, to ask questions, and I look forward to facilitating getting you all in touch with each other. And if any of you have any thoughts or questions or ideas about how we could better facilitate your interactions with each other, we'd love to hear them. As I say, this is an experiment in collaborative science that we are all in together, and so we really look forward to watching how it progresses and what the outcomes are. So thank you all again, and have a good day, and best of luck with your modeling.